Okay. Hi, everyone. So you all know me. Then we can skip the introduction part. Today I'm going to talk about iPSCs and how we use them in disease modeling and how it can be used in therapeutic development or therapy development. And maybe I should start by saying that actually I know many of you here work on iPSCs more than we do actually in our lab. So I feel like I'm not the best expert here, but I'll do my best to tell you about these and how we use them. I'll tell you a bit about what iPSCs are and start with the history of iPSCs. This audience probably doesn't need the definition, but let's start. Induced pluripotent stem cells are somatic cells programmed into a pluripotent state capable of differentiating into any cell type in the body. And as you probably all know, this story started with Shinya Yamanaka discovery in 2006, where he basically showed that introduction of 4 transcription factors, as we commonly know nowadays as OSKM or Yamanaka factors, can induce pluripotency in mouse fibroblasts. I like to, when I talk about concepts, I like to give some historical background first. So I hope it's not too boring for you, but I think it puts things into perspective. Actually, it did not start out of blue. So when Yamanaka made his discovery, he didn't go to sleep one night and came up with the idea, right? I mean, everything in science builds on discoveries that were made by others previously. So we can trace this story back to 1950s, where certain frog experiments were done to show that actually reprogramming is possible by nuclear transfer. And actually, the first successful demonstration was done in Xenopus eggs, and the transfer of somatic cells, nucleus, into. Okay, it's too weak, I think. Sorry. Let's see. Okay, let's do it this way. Şöyle yapalım bir dakika. Ha, bilgisayar göstereyim mi? Şuna yapalım. Okay. Yes, so it was shown that when, and I believe it was an intestinal cell from frogs were transferred into, nucleus from the cells. These cells were transferred into UV irradiated oocytes in frogs. It was able to generate viable and swimming tadpoles and frogs eventually. However, the efficiency was very low. And this basically shows that this is the first successful generation in early 1960s, which, as opposed to previous experiments in 1950s, it showed that actually a small percentage of these frogs are viable and can actually swim. As you notice here, not all survive. Okay, so this is only the beginning. Okay, in frog in 1962, John Gordon, he's the one who basically did this experiment. Actually, 3 streams, 3 roads led to the discovery of iPSCs in 2006. So it started, as I said, in the 1950s and 60s by these frog experiments, which followed in 1990s with the famous sheep Dolly. That somatic cell nuclear transfer can be done in mammals, actually. And another road was that, actually, I have some text here. Yeah, second road showed that actually certain transcription factors exist which we can call as master transcription factors, that basically determines and induces the fate of a given lineage. So this was actually maybe even more of an influence on Yamanaka, that showed that actually if you introduce certain transcription factors like MyoD induces myogenic fate, these introducing certain transcription factors can differentiate cells into certain fates. And obviously, practically, to be able to culture these cells, Yamanaka also benefited from studies that were done in mouse embryonic stem cells, which basically defined the factors that are required or that are that create the optimal media for stem cells, basically culturing. And these discoveries culminated in the shared Nobel Prize by John Gordon, I don't know how to pronounce his name, and Shinya Yamanaka in 2012. I mean, so they discovered this in 2006 and got the Nobel Prize in 2012. I included some quotes from Yamanaka in his presentation from his Nobel lecture, because I think majority of the audience here is students, young researchers. So there are some, I think, I really recommend you to read his or watch his Nobel lecture. There are some very teaching parts which I liked his. So one of these is that I put is that many laboratories working on ES cells, including those famous in the field, were working on in vitro directed differentiation of cells into various lineages, such as cardiac myocytes and neuronal cells. I do not think it would be wise to compete with these laboratories, since my lab was very small and new. I decided to do the opposite. So I think the take-home message here is that be bold. Don't try to stick with the mainstream always. Sometimes it's especially if if you do not have the resources with those big guns in the field, it might be wiser to try new things. So how did he do this? I don't want to take too much time here, but I think it's worth storytelling. So he started with 24 transcription factors, which he identified based on all his previous research and of research of others. And when he introduced these transcription factors individually, he didn't get any pluripotency in mouse cells. But when he introduced all together, he achieved, to his surprise, he says very openly that he didn't expect it to work, but it worked. So it worked, and he got some pluripotent cells. So the next step was like, which one of these 24 are really responsible? So he started eliminating one by one. So take one out, and then reducing it to 10 factors, take one out for each, and then eventually he reduced it to minimal number of transcription factors, as we all know now, Yamanaka factors. Yeah, so the next step was that, okay, now we did this in human mouse, but for years in the field of embryonic stem cells, the researchers struggled with making progress in mammals and humans. So it wasn't straightforward. And however, with all the knowledge that accumulated, he quickly showed that he quickly applied this approach to human cells, human fibroblasts, and he obtained, so these were from synovial tissue and neonatal foreskin, and he showed that the same factors can achieve pluripotency. So far, I checked, it received 25,000 citations. So now we know the road from to iPSCs, but what about from in the following years? I put this diagram here because it shows how quickly it developed. As you see here, in a year, major milestones, major studies were published that made such quick progress. So basically, the field embraced this approach so much that a huge progress was made in a few years. Yeah, and one of them was that it was shown by the group of Rudolf Janisch, that actually this approach can be used for treatment of sickle cell anemia. And in this study, basically, they use a humanized model, mouse model, obtain fibroblasts, tail tip fibroblasts, converted to iPSCs. And after correcting these mutations in these iPS cells, I want you to notice that this was in 2007, before CRISPR. So this was all done using the conventional gene editing technologies, and everything was done like in a year or so. And they showed that these cells, when they differentiate into amyloid bodies and then to hematopoietic progenitors, they can be introduced back to the mice and has the potential to cure. Other studies, many of them followed. I don't want to mention every single one here. It will take too much time. But so I put this also because it showed that this one is from 2008, and already just one group. So from George Daly from Harvard, he already published all these lines from different diseases. Many of them are genetic diseases. Well, I mean, monogenic diseases. And then it was shown that this can be applied to generate disease models, obviously. So the iPS, yes, the main goal is to use it as a cure or therapy, but it also proved to be really useful to model diseases, for many of which we did not have any really physiologically relevant diseases, basically. Okay, from early days to today, what changed? I skipped ahead because of all these studies that in the meantime, we cannot mention every single one, but I just checked the most recent clinicaltrials.gov website and found that if you search for iPSCs, 192 of them comes up. 43 of these are completed already, and 75 are recruiting. And this is one of the most recent ones. Maybe you saw recently in media, social media, or papers. Basically, this shows a type 1 diabetes patient receiving chemically induced, by the way. So this is not the original way of making iPSCs, but chemically induced iPSCs can be differentiated to islets under abdominal anterior rectus sheet, and it provided at least some level of success. So, okay, some other examples. Again, I just selected a few. I might be biased, but this one is with Parkinson's. So a company named, I think, Aspen, they are developing a iPSC based therapy to differentiate into patient-specific dopamine-producing neuronal precursor cells before transplantation into patient's brain. And this is this study recently started, so it received an accelerated status from FDA. So it's currently I think recruiting patients. Many of these studies, I just picked a few. So these, I selected ones so that are not all neurogenetic disease or neurologic disease. So it can be used in certain cancer types to generate like natural killer cells from iPSCs, autologous cells. Or one of the actually most well-known examples, one of the earliest success stories in actually iPS-based therapies is retinal pigment epithelium cells, RPE. So it's one of the first application areas of iPSCs in disease treatment. So many of these studies are going on, but not all studies with iPSCs aim to cure diseases. Actually, in clinical trials website, you can find many of them that actually aim to develop iPSCs as disease models, as you can see here, or biobanks to generate disease models later on. So this shows that iPSCs in clinical studies are being used as to generate disease models too. Some of them actually try to bring together the genetics actually to make predictions about health or disease course or to develop treatments, basically to combine genetics of the patients together with iPSC models. And these two are related to the cardiovascular diseases. I put two of them here. Actually here, Esra Hocam, when in her talks, she always talks about these Modernization Act and how we use organoids, organoid on chips. So many of you I know are in the Erdal lab. You must be very familiar with all these concepts, but for those of you who are not, FDA Modernization Act 2.0 actually it paved the way for computational biology and clinical trials in a dish. So those are two main concepts. And the current paradigm, maybe, or flow diagram, we can call it, is basically nowadays we are using AI to predict drugs or even develop novel, non-natural, non-existing novel molecules, basically come up with like peptide drugs, antibody drugs, vaccines, RNA-based therapeutics, antisense oligos, small molecules. And with all these new computational power we have, we are coming up with really novel molecules and testing these in vitro in more traditional methods. We can go either to the small animals, which are the traditional way, or if fast track is possible, then actually when animal studies are not relevant, iPSC, these large-scale clinical trials in iPSCs using either organoids or organ on a chip or organoid on a chip technologies, we can now perform these clinical trials first in iPSCs, which provide a lot more relevant model compared to 2D cultures and offer many advantages over animal studies. So after this step, now we can go to the clinical trials and achieve higher success. Obviously, there are many challenges that needs to be overcome. So with all these promise and hype, we know that there are still many challenges. So the challenges associated with applications in biomedical research is either to model diseases for etiology or pathophysiology, or for personalized and precision medicine applications, as we just mentioned. Some of these include basically derivation of clinical grade iPSC-derived therapeutic cell products for acute life-threatening conditions. So this is not as straightforward as it may seem. And acceptable quality criteria of iPSC-derived therapeutic cell products need to be defined. So this is still a gray area. We still need to clarify these. Tumorigenicity is still a problem, obviously. So you cannot just, I mean, one of the criteria actually for iPSC confirmation is to inject into mice for teratoma formation. So it's that tumorigenicity must be controlled really well. Immune rejection is still a problem. So phenotypic heterogeneity is a major problem. Yes, now we have much better differentiation protocols, but still we have a way to go. So iPSC differentiation to different cell types still needs to be improved. So inefficient recapitulation of late onset diseases due to lack of maturity. So yes, we can model certain diseases that are that usually manifest or that usually pathogenic changes that occur early in development. We can model them in iPSCs relatively easily, but for those that appear later in life, those are still a challenge. So one of the major challenges obviously is to generate large cohorts of iPSC lines. for polygenic and sporadic disease etiology, because these are not monogenic diseases. That means every single iPS line coming from different donors are different. They have different genetic background. So we need large cohorts, but the problem is that it's too expensive. So you cannot just generate iPS lines very easily and cheaply. And genetic instability, obviously, this is a concern that always needs to be addressed. But a lot of sharing is being done. So iPS repositories exist. So if you want to study certain diseases, so this is from 2018. It's not the most updated one, but still it's a useful one. You can, if you want to study certain diseases, you don't need to generate iPSCs yourself sometimes. You can just go and purchase or collaborate with researchers and get these lines. So this diagram I like because it's kind of conceptualizes like what kind of steps needs to be taken care of when we develop drugs or therapies using iPSCs. So in the first step where cell programming is performed, some of the steps include like a generation of master bank, cell bank, quality control for pluripotency, sterility, and lack of genomic instability or integration. These are all really critical. And then you move on to having a working bank where you can perform further quality control steps. And next comes the differentiation. Okay, you have good iPS cells, but what about differentiation step? You must have protocols that are really robust and that minimize heterogeneity. And you must validate them. You must characterize them by percentage purity at molecular level, functional level. So these all a lot of work, basically. And then you have another working bank, which you still need to further do quality control by using different molecular and functional assays. Okay, next step is assay design. So you have the differentiated cells, but what kind of assay you can perform to really use as readout? So it must be translatable. So cell type here is important. If you are modeling a disease, so you must be differentiating your iPS cells to a relevant cell type to really have a valid and relevant readout. Then you must validate this assay in isogenic cells, also in different lines. So one line is not enough. And then there's technical assay validation where you basically look for positive and negative controls and reproducibility. So these are really important. And when you do screen, for drug screen, you do the primary screen and you perform counter screens or followed by orthogonal assays and secondary assays. So this may be more common to other types of models, cellular models, but still needs to be done. And then you move on to lead optimization. Most of this is also true for actually for when you want to do just disease modeling too. It's not only for therapy or drug development. So you still need to perform similar quality control and similar workflow. You need to develop similar workflows. I don't want to go into too much detail for the sake of time, but I'll be happy to discuss. So as we said, heterogeneity is a major problem when you model diseases. So if you have two different lines from different laboratories, you might have, when you analyze the results, especially with omics approach, you can have results that needs to be properly handled with tools, statistic and bioinformatic tools that will really help you basically understand the real underlying mechanisms. As we said, consistency is a major problem. When you generate cells from iPSCs, you must have homogeneous cell populations. So there are many approaches to answer this need. One of those is to use certain transcription factors, at least for neural differentiation, if you transduce cells with certain transcription factors like NGN2 here, and generate these NGN2 expressing MP neural progenitor cells, which can be induced by actually doxycycline, then you will have at the end neural cells that are a lot more homogeneous, basically. And this basically shows the main steps, but I'll mention about these when I mention about our own study. So I'm gonna skip here. Yes. So let me mention a bit about what we have been doing, how we are using these iPSCs. So this is from a study where our lab with Professor Semra Hız, we basically identified with our collaborators a novel mutation, novel variant in a gene named CATNAL2. So this CATNAL2 is a member of catanol family, but before that, let me mention quickly. So this is an 11-year-old male patient with epileptic seizures, intellectual disability, autistic behavior, and dysmorphic features. And when we did the segregation analysis in the family after whole exome sequencing, we confirmed that this is a good candidate. So this variant is, it resides in this AAA ATPase domain of this CATNAL2. So a little bit about this family. So these family are a family of enzymes that or proteins that basically spheres cuts microtubules. Okay, there are like 5 or 6 members, and they basically function as heterodimeric complexes, and they are known to play roles in cell division, migration, signaling, and homeostasis. Yeah, basically we asked the question of how we can basically understand the effect, functional effects of this variant on these patient cells. We started with collecting fibroblasts, dermal fibroblasts from the patient, and Dr. Tamer Önder at Koç University, he used the episomal plasmid approach. So I didn't put a separate slide for this, but there are different ways to generate iPSCs, and episomal plasmids are one of the most commonly used ones. We generated iPSCs, actually he generated iPSCs, and we took these iPSCs from him and converted neural progenitor cells. I'll come to this in a minute, but basically this is the part where you collect skin biopsy and culture in regular media, let it grow. And after the introduction of these Yamanaka factors, wait for the colonies to appear, basically. It may not work in the first time, which it was the case for us. So he had to repeat it the second time. And he got several colonies eventually, and you see how they basically look. And on MEFs, on mouse membrane fibroblasts, a single colonies, and on mitoGel. One of the most important steps in iPS generation is obviously quality control, as you mentioned, and routinely markers that are used for pluripotency, like OCT4, SSEA4, TRA160, and SSEA1 as negative control. These are used to assess whether these cells are truly pluripotent. So here it was done by immunofluorescence. Another way is to check for lineage, differentiating in different lineages. I didn't put slides here, sorry for that. I thought I put, but you differentiate into 3 main lineages, and then look, stain for those lineage markers and see if they're able to really differentiate into these lineages. Then you also need to make sure that, if you remember from the previous slides, you need to make sure there's no integration into the genomic DNA. So confirming that there is no integration of these episomal plasmids into genome as expected. And you also need to perform karyotype analysis, because it's a not uncommon observation that some of these clones can really have karyotypic abnormalities. Okay, what we did, we start with fibroblasts first. So this shows how these catenins, catenal-2 work. We looked at certain proteins that collaborate, work together with catenal-2 in regulating microtubule dynamics. In fibroblasts, I just want to summarize very briefly. In fibroblasts, we saw certain defects with microtubule organization. However, it was not as clear-cut as we hoped for. Then we decided to convert them into neural progenitor cells. There are many protocols to use, actually. So I just put this slide here to show you can use similar but distinct different protocols that use like here, as you can see, like dorsomorphin with this small molecule can be used both for cortical neuron protocol and also motor neuron protocol, but the timing is different, so they're not all same, very similar, but you need to be very accurate with these protocols. We used a very commonly used commercial kit, STEMDiff. So it's a SMAT inhibitor neural induction kit and a neuroprogenitor medium. Basically, this kit uses dual SMAT inhibition approach to inhibit differentiation in certain lineages and puts them toward neurogenic fate, directs them towards neurogenic fate, and this is how colonies look through these differentiation stages. And next step, you need to confirm that they are really neural progenitor cells or neurons, whatever you like to do. But here we convert them into neural progenitor cells and confirm certain markers like nestin, SOX1, PAX6. These are all markers of neural lineage and absence of OCT4 as a pluripotent marker because they must lose OCT4. And next step was to analyze these MPCs using all these markers that I just mentioned that work together with catenal. Long story short, we observed a much more severe microtubule organization defects in neuroprogenitor cells compared to fibroblasts, which actually is something that we expected, but it was nice to see. Fibroblasts are okay to some degree to model disease, but to see the real effects, sometimes you really need the most relevant cell type. And we saw that these MPCs showed these defects as expected. Next thing, you can do many things. You can do function assays, omics analysis. I'll skip through. We did proteomic and RNA sequencing. In this example here, you see proteomics characterization, and we performed some rescue experiments on the cells. So basically, you can do rescue in two ways. The ideal way is to obviously do CRISPR correction of the mutation, but it's quite challenging. So we chose a more practical approach where we introduced the missing normal copy into these cells with a tag, and then looked for differences. So we did this as transient transfection, and we had some interesting observations, which is not the real topic of today, so I'll kind of skip these. Sorry, I forgot to mention all these studies, were done thanks to our close collaboration with pediatric neurology department here, Dr. Sam Rahu's crew. So when you want to do such disease modeling, it's important to have close clinical collaborations, obviously. I'll mention just by a few slides about many of you here work on CRISPR. So I'm not going to teach you what CRISPR is. You already know, I'm pretty sure. But CRISPR genome editing can be used in disease modeling. So if you do not have access to patient cells, which is the case for many times, in many cases, you can generate a mutation yourself, or you can correct the mutation. So you can do both ways. The original Cas nucleases were basically, they create double-stranded breaks, and you can disrupt the genes, which we do very, it's much easier to do. But the bigger interest here is obviously in correction, so more editing, like at base level. So base editors came out later on, and prime editing was another nice introduction to the toolbox of Cas toolbox, and we can correct mutations using these. But the main problem here again is the efficiency. And when you deal with like iPSCs or stem cells, which are quite how should I say, not as easy as cancer cell lines, it's a bit challenging and expensive. You can use, if you are not interested in generating a mutation, if gene regulation increasing or decreasing the activity of gene is enough for you, can use other CRISPR tools that allow you to just increase or decrease the expression of genes. So I'll mention very quickly here how we use CRISPR in our studies. We are working with Genç Pınar and Semra again on cortical malformations, pediatric cortical malformation, and especially the lissencephaly type. So cortical development is a highly regulated process, and this relies heavily on neuronal migration and cell division. So certain genes, when they're mutated, they affect these either generation of new neurons or migration of neurons, so which leads to loss of typical gyrus and typical structure of the cortex. Here, instead of using iPSCs, we worked with NPCs, neuroprogenitor cells, because it's cheaper and more practical. So from iPSCs, we convert NPSCs from iPSCs, and using a microfluidic chip named IC chip, which basically where you put two cells in one chamber and put two serum in the other chamber, and with matrigel in the middle, you allow the cells to migrate toward the serum basically. And when they enter through these channels to the matrigel, you can follow them and quantify them and see if there's any defect. So that's what we did. And then we did this for many genes. I don't want to show everything here, but basically for some of them, we saw that when we quantified that, targeting this gene actually, where we use two different colors, basically we put them in the same chamber, targeting these genes cause some migration defects, at least for some genes, not for all, but this is another example of how you can do it. You can also do electrophysiological analysis, but you need to have access to these electrophysiology equipment. Many of you here work with organoids. I don't want to take too much time talking about them, but nowadays, it's from 2019, again, not the most current one, but there are many organoid models. So actually, that's like now the mainstream of actually developing disease models. Or now we are moving on to organoid on chips, basically. So you can also model not only these monogenic diseases, but also sporadic diseases, which are mostly polygenic, actually. So here you can, with the same approach, you can activate or deactivate genes using CRISPR, like with rather than mutating cells. Another approach that's useful for this purpose is also useful for monogenic disease too, but when you convert cells to iPSCs and later to your relevant cell type, which could be neurons, this takes a lot of time and a lot of money. So direct programming offers an alternative. Here you can introduce certain transcription factors, and in a very controlled media, you can achieve direct conversion. Honestly, we tried this, but we couldn't achieve. But maybe if it's right enough, maybe we would achieve. So I mean, it saves you a lot of time and money, but it's not possible for all cell types, obviously. So disadvantage is that it's less controlled. So at the end, the cells that you get is less homogeneous than what you get from iPSCs. But still, it's something. Yeah, you can use these cells to do drug screens, as we mentioned already. Okay, I would like to wrap up with, again, some wise words from Shinya. I'll just read it. In 1989, however, my life took a new turn from clinical medicine, orthopedic surgery to basic science research for two reasons. First, I found that I was not a very talented surgeon. So this is something many of us might feel at some point in our life. So even a Nobel laureate can feel this way. So second, I saw many patients suffering from intractable disease and injuries, which even highly talented surgeons and physicians were not able to cure. For these reasons, I decided to go back to school. I became a PhD student at Osaka State University Medical School in April of 1989. So it's never too late to go after your dreams, I should say, maybe. The generation of iPS cells was an exceptional experience in my scientific career in that everything went smoothly. In all other cases, my career has been full of failures. So this is another lesson to, I think, to remember. I'd like to thank our collaborators here at IBG, my IBG lab members, obviously first, and then our collaborators at the university and at IBG. And thank you for your attention.